Okay, we're back. Biology of cells and tissues. And we are diving into blood vessels. As always, again, leave a like on the video, subscribe if you haven't already. Helps out a ton. So, blood vessels in general. We saw this slide already. Arteries, veins, capillaries, that's what we're talking about today. So arteries carry blood away from the heart. They get progressively smaller. So they start off big, get smaller. Veins carry blood towards the heart, and they start off really small and they get bigger as they go towards the heart. And then capillaries is where we'll spend actually most of our time. So in general, there are 3 layers seen in our vessels. Specifically, the veins and the capillaries is what we're going to be mostly talking about here. We have these 3 layers: tunica adventitia, tunica media, and tunica intima. Tunica adventitia is also known as tunica externa. Tunica intima is also known as tunica interna as well. And then we'll talk about another structure known as the vasovasorum, which actually should be here. Okay. So the tunica adventitia is the outermost layer. It's made up of type 1 collagen fibers as well as elastic fibers. It is continuous with the connective tissue of the surrounding organs, so it helps, layer everything, keep everything in place. And part of it, some of our larger vessels, especially the veins, have a part of it called the vasovasorum. This is what provides nutrients for the outer layers of the media and the adventitia of some of the large vessels. And it's more frequently found in veins because if we think about it, within the veins, they don't have the oxygen-rich, nutrient-rich blood because they are getting the deoxygenated blood on their side after it's already gone through all of the tissue. So they need a little extra help to make sure they get all the nutrients that they need. So here we can see artery versus vein. Vein has a little bit of a thicker tunica externa, and it has all of those vessels, that vasovasorum. Vasovasorum is literally just meaning vessels within the vessel. The tunica media, this is the middle layer of the blood vessel. You have these layers of smooth muscle, concentric meaning circular, so they go around the vessel. It also has a bunch of elastic fibers, reticular fibers, and proteoglycans. This layer is big, is responsible for vasoconstriction, vasodilation, as well as returning the vessel to its original shape. Especially having a lot of elastic lamina in larger arteries. Because you think about arteries, especially, arteries have a lot more pressure and they tend to expand and they dilate and they constrict because they need to be dealing with pumping the blood through them throughout the entire body, throughout the systemic circulation. So we need a lot more muscle, we need a lot more elasticity. I should say more elastic fibers that help return it back to its original shape. We also have the tunica intima, also known as the tunica interna. This is the inner lining. It is endothelium, endo being inside, thelium, endothelial cells, epithelial cells. There's also some subendothelial connective tissues, and we have an internal elastic lamina as well in the arteries. Here are our different layers: tunica intima, tunica media, and the tunica adventitia. This picture is really, really good. I have a couple of graphs later, a couple of charts a little bit later on that kind of really cover a lot of the differences. Specifically, this chart right here is really good. This compares the different layers and the different components of arteries versus veins. Big things to note. In arteries, the tunica media is much, much larger than in the veins. And the veins, the tunica adventitia, is larger than in arteries. That's like the main thing that you get asked about. So talking about arteries themselves, again, these have the thicker walls compared to veins. More tunica media. They transport blood from the heart to the tissue. They resist blood pressure, changes in blood pressure in the larger arteries, and they regulate blood flow in the arterioles, We'll talk about what arterioles are in a minute. So we have large arteries, medium arteries, and then small arteries or arterioles. So our large arteries, these are our elastic or conducting arteries. They have that thick tunica intima, thick tunica media. They have what's called a fenestrated elastic lamina. Fenestrated means having one or more openings or pores. It has little holes in it. And they have many layers of smooth muscle. Here's what our large artery, this is an elastic artery. This is what it looks like. So that massive tunica media layer, thinner tunica adventitia, and then a thin endothelium. Medium arteries, these are distributing or our muscular arteries. The tunica media has a lot more smooth muscle and less elastin. It's prominent internal and external elastic lamina. These are kind of bullet points. These are the kind of things, honestly, throw these into a little Quizlet. Identify, is this large, medium, small arteries, large, medium, small veins, and go from there. Okay, that's what you're going to see. There's our medium arteries. Here's a picture of our medium arteries right there. And then we have our small arteries and arterioles. The tunica media in small arteries is 5 to 10 layers of smooth muscle, 1 to 4 layers in arterioles. So kind of the order that we see in size is we go from artery, large artery, medium artery, small artery, arteriole, then to capillaries. Then we'll see on the venous side, venule, small veins, medium veins, large veins. Here's a picture. We have a small artery and a small vein. You can clearly see the difference. You can see the blood cells inside of there. Small artery versus the vein. And notice how the artery is much more circular and veins tend to be more oval shaped. That's because of that muscular layer. The muscular layer helps it retain that circular shape, whereas the veins tend to get a little bit more squished. Now talking about the veins. So veins return blood from the tissue to the heart. They have a lower pressure than the arteries. They have thinner walls, larger lumens. Lumen just refers to the hollow space inside. Overall, they have larger lumens than corresponding arteries. They have endothelial covered valves to ensure one-way flow. And then again, we see a large, medium, small, and then venules. Here are what those valves look like. So we have these unidirectional one-way valves. So blood gets pushed through the veins. Most of the time, the way that we actually get blood back to the heart is through muscular contraction. The veins are running in through a lot of our muscles. When our muscles contract, it kind of squeezes the veins and pushes the blood up, and then it can't flow backwards because of the valves. So it pushes, our muscles push it towards the heart, and then it can't go backwards. So that's how a lot of our venous system works. We have large veins, a good intima, a thin media, and a thick tunica adventitia. Again, that thick tunica adventitia because we need to have more vessels and stuff running through it to feed that tissue. Our small and our medium veins have thin tunica media, well-developed tunica adventitia, the same, and they have those valves. This is not to say, so the larger ones do have the valves, but the ones that we most commonly observe, we observe more the medium and the small ones having the valves. Finally, the venules, these are our post-capillary venules. They are similar in structure to capillaries, but they have a larger lumen. Again, that's that hollow space on the inside. And they have a similar function to capillaries. So they're still working a lot with, our exchange of nutrients, exchange of fluid, all of that kind of stuff as the capillaries do. We have a bunch of pictures here. Venule versus arteriole. Arteriole, again, thicker, much more round. Venule, more oval shaped. There's the vein. There's the artery, again, thicker on the outside. artery. There's a medium-sized artery, medium-sized vein. Thicker, that tunica media, versus this one, much more round, irregular shape. Large veins do have a muscular media layer, the tunica media, but it's still very thin compared to the surrounding adventitia. So here we have the intima, the media, and the adventitia again. This right here, this is an example. This is one of those valves where it'll let blood through. This looks like the blood most likely is going this way, because blood's going to come this way. I can't see the full picture, but blood's probably coming this way, and then it prevents backflow this way. Here's that size chart. Again, a lot of this, a lot of information that you want to focus on is right in here, kind of comparing the different layers, which has the more adventitia, which has more media, which is what's the lumen size, the overall thickness. That's kind of the main things we're going to get tested on here. And then just like the general structures, like what is the tunica externa, what is the tunica media, what is the tunica intima, what are those structures, what's in each of those. So that is our arteries and our veins. Now capillaries. Capillaries, this is where we spend a little bit more time, because there's some very important things about capillaries, and there's a little bit more difference between the types of capillaries. So capillaries have a diameter of 7 to 9 microns. That's very small. Again, a lot of capillaries are even smaller than red blood cells themselves. That's why those red blood cells have that specific protein to let it bend and form into that shape. Don't remember the name of that protein right now. I think it's... Make sure I say it right. Spectrin, that's it. Spectrin, they have that spectrin protein that allows them to be flexible. Okay, the capillaries are often just a single layer of endothelial cells or epithelial cells resting on a basal lamina. They're held together by tight junctions. If you don't remember what a tight junction is, think back to one of the lectures at the beginning of all of this material. We talk about those junctions. They have these structures called pericytes. These are kind of like replacement cells, protective cells. They wrap around, and if then you get any damage, these pericytes can kind of go in, cover up the damage, and actually become part of the wall and differentiate into endothelial cells. This is after tissue injury in order to rebuild the vessel. So our pericytes. And we have 3 specific types of capillaries: continuous, fenestrated, sinusoidal. If we think about those words, continuous, meaning that there are no breaks. Fenestrated, we talk about having pores or holes in it. And sinusoidal, sinusoid, is referring to larger gaps in there. So in general, we have the endothelial cells on the basal lamina. Those are those pericytes. The continuous capillaries, these are found mostly in our muscle, the lungs, connective tissue, exocrine glands, and nervous tissue. Throw those into a Quizlet. It's good to know all the locations. It has no pores because it's continuous. And the way that it actually transports things in between cells is called pinocytic vesicles for cellular transport. I don't worry too much about that whole process, kind of what that is. Just know the name. Know the name that it's pinocytic vesicles. Here's just another picture of our continuous capillary. Continuous, no, not really a whole lot of holes or anything in between the layers of the epithelial cells. Endothelial cells, sorry. You should say them properly. Fenestrated, these have fenestrae or pores, and they still have a continuous basal lamina. This is where rapid exchanging occurs. Places like the kidney, intestines, endocrine glands, where you really need a lot of stuff moving through at a time. So rapid exchange. Another picture. A blood cell fitting through there, the holes, the fenestrations. That's the way stuff can get through it quick. Finally, sinusoidal. These are larger than our fenestrated or continuous capillaries. These have big gaps, they call them intercellular gaps, between the endothelial wall, these big old holes, as even the basal membrane, the basement membrane, has holes in it. And this is because sinusoid is located in the liver, the bone marrow, and the spleen. And these specific locations, if you think about, this is where our red blood cells need to be able to enter capillaries and enter vessels, or be able to even leave vessels. The spleen is responsible for breaking down our old red blood cells. Also the liver does a lot of detox of the blood. So there's lots and lots and lots of blood held within the liver, as well as the liver is responsible for maintaining like our blood glucose and that kind of stuff. So we need to be able to have blood cells and stuff coming out and going in easily through those capillaries. So that's why they have these bigger gaps, these sinusoid capillaries. Another couple of pictures, and this is just what they look like each individually. Things to remember about these, you'll want to remember, which one has holes, if it's continuous or not. The basement membrane, the continuous fenestrated have a complete basement membrane. Sinusoid has an incomplete basement membrane. I don't know why that arrow is following us, but and then you'll want to remember the locations of where these are. The continuous being in the muscle and the lung tissue especially. Fenestrated being in places where you need a lot of transport. Endocrine system, the kidneys, the intestines. And sinusoid being where red blood cells need to be able to enter and exit. So places like the liver, the pancreas, not the pancreas, the spleen, the liver, the spleen, and then the bone marrow being in the sinusoid. And that is our circulatory system lecture. Of course, if you want to get access to the notes, as well as if you have not gone back and re-downloaded some of the material recently, as I go through these PowerPoints and revamp them for these recordings, as well as doing the recording, I add little tidbits. I do go back and replace those files, so make sure you go back into the Google Drive and re-download the new PowerPoint, so that way you have the most updated information. Thanks everybody for watching, and have a wonderful day, and good luck studying.